In this module, we discuss contamination control in the special context of pharmaceutical manufacturing. Pharmaceutical manufacturing is governed by good manufacturing practices which seek to safeguard the safety, efficacy, quality, or safety, identity, strength, purity, quality of products that are placed in the market so that patients are not placed at undue risk. Amongst many things that could affect safety, efficacy, or quality, or adversely impact upon the identity, strength, or purity, one thing certainly is contamination. Contamination could compromise the purity and alter the identity of the product that is being offered to the patient, rendering the product both adulterated as well as misbranded. And so you can be very sure, depending on the level of risk you may place a patient at, any regulator visiting your facility is going to put your contamination control measures and program under the microscope. Contamination control is a quality management subset. Pharmaceutical quality management recognizes that any process has inherent to it the risk of rejection. Variability in a process is intrinsic to that process, and pharmaceutical quality management seeks to put policies backed by procedures in place to try and control that variability and minimize the risk of rejection, and do all this to acceptable levels. You cannot eliminate variability, so you can only control it, and how much to control it will be determined by the specifications that govern the product. I'll give examples. If you are making a pharmacopoeial formulation, the pharmacopoeia tells you what is acceptable quality, what should be the assay, and what should be the range within which your assay falls. Those making sterile products have been told that their non-conforming units, the defects produced, shall not exceed 0.1%. When it comes down to sterilization, you're told that the sterility assurance level should be 10⁻⁻⁶ or better. These are the kind of acceptable limits that are available to you, and you have to then set your own internal limits. If you are an API manufacturer, you state what are the process-generated impurities and what would be considered acceptable. Now, if it is 1000 ppm or less, ICH Q7 tells you not to bother. But if it is more than 1000, please characterize it. And if it is more than 5000, please not just characterize it, but also carry out special studies if required to establish that the safety has not been compromised in any way. The toxicity is within acceptable limits. But this relates to process-related impurities, residual solvents and related substances. But there's one more thing that we all worry about. That is the carryover of product A into product B. The acceptance limits we set for our cleaning, and which is what we validate. There they tell you, please look at the pharmacological aspect of product A in product B. So you set therapeutic limits. You look at the toxicity of A and the cleaning and the residues going into B. And finally, a generic limit like 10 ppm. You calculate the therapeutic limit, calculate the toxicity limit, and compare it with 10 ppm. Whichever is the lowest is what you set as your acceptance criteria during cleaning validation and the subsequent cleaning that follows. Likewise, contamination control is about recognizing that with contaminants so ubiquitous in the environment, the risk of contamination is ever present. And therefore, we try to work out ways and means, put policies backed by procedures in place with a view, if not to eliminate contamination at every stage, but at least to minimize the contamination to acceptable levels. Contamination control, I repeat, is about putting in place systems, practices, procedures to minimize the risk of contamination.